Hello everyone, my name's Sarah. I'm a postdoc at the University of East Anglia, and today I'm going to be talking about gut microbiome variation and senescence in the Seychelles Warbler. So senescence can be defined as a decline in function of physical systems with age, and it occurs in most animal species. However, the extent to which the gut microbiome undergoes senescence remains unclear. So to give you some background, the gut microbiome is a diverse microbial ecosystem made up of bacteria, microbial eukaryotes, and viruses, many of which contribute to fundamental host processes, including things like aiding digestion to providing the host protection against pathogenic infection. And there's been lots of research showing that the gut microbiome can be highly dynamic over time within an individual. So it's responsive to things like dietary changes, seasonal changes, and the onset of disease. However, the extent to which the gut microbiome shows consistent changes with age and shows signatures of host senescence remains unknown. There are several reasons why we might expect the gut microbiome to senesce, particularly if other host systems are senescing, for example, the immune system, which plays a really integral role in determining which microbial species can actually colonize the intestinal tract. So if the immune system is senescing, the host may become less capable of maintaining a diverse, beneficial community of microbes, particularly if it also becomes less able to screen out and prevent the proliferation of pathogenic species. And as an increasing imbalance in the number of beneficial and pathogenic species occurs, we might expect that the beneficial functions normally provided by the gut might decline. And this could in turn feed back to influence other host systems and cause a reduction in host health. It's also important to note that the rate at which this process occurs and the resilience of the gut microbiome to senescence in other host systems might be a source of variation affecting the rate at which senescence occurs across individuals within populations. So there's been lots of interest in whether the gut microbiome shows signatures of senescence. And there have been several studies carried out in humans and laboratory organisms that have shown that there tends to be a gradual reduction in microbial diversity in the gut with age. But there's also an increase in the abundance of pro-inflammatory microbes, which are often linked to an increase in gut leakiness and the onset of age-related diseases. Interestingly, though, they've also shown that individuals that reach extreme ages, such as centenarians, tend to have a greater number of beneficial anti-inflammatory microbes in their gut. So there does seem to be quite a lot of variation across populations in the extent to which the gut microbiome changes with age. However, the extent to which we can generalize from these studies to other systems, particularly wild systems, can be quite limited for a number of reasons. So particularly in human studies, there are lots of confounding factors that are associated with age that also influence the gut microbiome. So for example, elderly people are more likely to be taking medications. They may also enter permanent care. And we know that living in care homes can significantly alter your gut microbiome. So it might be that the changes that we see with age are actually more to do with lifestyle factors changing than an intrinsic senescence process. There are also issues with studies looking at laboratory organisms because we know that lab animals have very different microbiomes compared to their wild counterparts, partly due to reduced genetic diversity, but also they're exposed to much simpler environmental conditions. A final issue with many of these studies is that the majority of them are cross-sectional, so they compare the microbiome of older individuals to that of younger individuals, but don't look at changes within an individual over the course of its lifespan, which is really important for identifying senescent patterns. So we hope to see whether we could identify senescent patterns in the gut microbiome of a wild species by looking at the Seychelles Warbler population living on Kuzan Island. And this system is useful for studying senescence for a number of reasons, one of them being that we have individually marked most of the individuals on the island, so we can get really good longitudinal measurements from the same individual over the course of its lifespan. Secondly, we have birth dates, so we can know how old each individual is, and we also have death dates, so we can look at changes prior to death. Previous research on this system has shown that this species does undergo senescent declines, so individuals older than 6 years of age tend to undergo declines in survival probability and relative reproductive success. And we wanted to know whether the gut microbiome showed similar signatures of senescence. So to do this, we collected fecal samples since 2017 and extracted the DNA from these fecal samples and sequenced the 16S rRNA gene to look at the bacterial component of the gut microbiome. And when we do this, what we see is that the gut microbiome is dominated by proteobacteria, firmicutes, and actinobacteria, but the extent to which these phyla are present varies quite substantially across individuals within the population. And what we wanted to know is whether any of this variation was linked to host aging and senescence. So in particular, we wanted to know whether the diversity of the gut microbiome, so the number of species present in the gut microbiome, changed significantly with age. And secondly, whether the composition of the gut microbiome changed with age, and in particular, whether it differed between pre- and post-senescent individuals. So first of all, to look at diversity. So we found quite a substantial reduction in diversity with age. So here on the x-axis is age in years, and on the y-axis is KO1 richness, which is an estimate of the number of bacterial species in the gut. Note that this has been log transformed, and a reduction from about 6 to 5 is a loss of about 300 bacterial species. So there was a significant reduction in the number of species in the gut with age in the warbler. So the next question is, what could be driving this change? And as I mentioned earlier, it could be linked to the senescence of other host systems, for example, the immune system. But it's important to note that it could also be linked to selective disappearance, so the loss of individuals with high levels of diversity in their gut from the population. And to distinguish between these two explanations, we really need longitudinal data so that we can look at within individual patterns. Unfortunately, due to issues with sampling over the past year and a half or so, we don't have enough individuals with multiple samples, particularly bridging that pre-post-senescent divide, to look at within individual changes in detail. But this is certainly the plan for the next year or so. So what about gut microbiome composition? So to analyse this, I first of all conducted a principal components analysis. So here each of the points is an individual, and how far apart the points are represents how dissimilar their gut microbiome is from one another. I then extracted the principal component axis scores for each of the axes in turn and related these scores to host age. And what I found was that there was a significant quadratic relationship between host age and the principal component 2 axis scores. So what this shows is that the gut microbiome becomes different from when you're a juvenile through adulthood until you're about middle age, so around the age of 5 or 6. However, after this point, the gut microbiome seems to stabilize and we don't see any further change in the gut microbiome. Now, obviously, again, we need to look at within individual data to see whether this matches what we're seeing between individuals. But at the moment, we don't see any evidence that there's a dramatic shift in the microbiome of senescent individuals compared to middle-aged individuals. And most of the change in the microbiome seems to occur gradually between the ages of about 0 and 6. So in conclusion, then, we found that microbial diversity declined with age in the gut of the Seychelles Warbler. And the gut microbiome composition also changed gradually between juveniles through early adulthood until they were about 6 years of age. After this point, it didn't seem to be that the gut microbiome changed any further, so it seemed to stabilize. And senescent individuals didn't have a dramatically different microbiome to those individuals that were middle-aged. As I said before, it would be really important to see whether within individual data matches what we are seeing in this cross-sectional data set. And because we are also interested in senescence, which is the decline in function with age, it will be really interesting to collect data on bacterial function. And to do this, we really need metagenomes and transcriptomic data. And we're in the process of collecting this and looking at this over the next couple of years. So with that, I'd like to thank everyone that's been involved in this project, and thank you very much for listening.